fibrovascular growth that grows over the cornea. And it is quite prevalent. I guess someone can just start the animation. Okay, awesome. So it's quite prevalent. So actually there's estimated to be about close to 1 billion people worldwide who have it. And in the US, about 15 million. Some of you may know it as surfer's eye or farmer's eye. And if you go to the South, California, Arizona, Florida, you'll see more people with pterygium. Pterygium is not just a cosmetic concern for people, obviously causing red eye, but it causes significant irritation. And in severe cases, can also cause astigmatism and loss of vision. And that's why when it causes vision loss, we remove it. But as an ophthalmologist, we try not to remove it because in a lot of time, or about 30% or so recurrence can occur. Recurrent pterygium is much, much harder than primary pterygium to treat. So as an ophthalmologist, even if it bothers the patient, we try not to remove it. And the major kind of gap there is that there's no FDA-approved drug for either primary pterygium or recurrent pterygium. So surgery is our only option. So my lab has had an NIH grant from the National Eye Institute to study the mechanism of pterygium, because so far we have only relied on kind of analysis from tissue, but what we were able to identify, and since IP is still pending, I'm not going to name the exact cells or molecules, but we were able to identify the upstream cells and show that UV light would activate this upstream cell to secrete a mediator to then act on the downstream cells. We're able to do experiments in vitro to kind of validate this, but the importance validation came from patient specimens. So we got about 17 pterygium and normal sense, and we were able to show that within the compartments of these cell types, that specific cell phenotype is activated in pterygium compared to control, both the upstream cells and the effector cells. So then we asked, well, why is recurrence rate so high? So then we said, why don't we look at the mediator? When we looked at that kind of mediator, we found that the mediator is very low in normal healthy conjunctiva, and in pterygium is very high. And in the tissue that we leave behind, we found that there is the mediator still present there. So we think that this is the cause of recurrence. So our approach, which probably many of you have guessed, is why don't we target the mediator, which is exactly what we propose here, is to target the mediator. And we figured that if we target this specific mediator, and it's not just one target, we obviously starting with the mediator, but we can target the downstream receptors on the effector cells as well. So we think this is kind of a good timing for to start a therapeutic for pterygium for a few reasons. Obviously, first one is we have kind of cracked the mechanism, we hope. And the second is topical biologic has recently been approved in the eye, on the ocular surface, so it has been de-risked. And even though there's no FDA-approved medication, there's actually a phase 3 by Cloudbreak, which actually is quite exciting, and I think it validates the market. I think the approach of targeting the upstream mediator would be more exciting than Cloudbreak's, which is a small molecule inhibitor, because they target just the downstream vessels, and that's why they cannot target recurrence, as their phase 2 shows. So the results have mainly relied on perception of reduced injection in the eye. However, we think that it's still very exciting that there's someone else kind of starting this. If it gets approved, at least will be a good kind of benchmark for us to compare against. And it also validates the market, the need to kind of study pterygium. So our lead indication is pterygium recurrence, because usually happens within 6 months, easily to diagnose it on the slit lamp. But also once, hopefully, if Cloudbreak's, let's say, gets approved, then primary pterygium study could also be easy to run. But beyond this, what we found is that some of these, the biology or the mechanism would apply to other corneal transplants, corneal neovascularization, or growth of new blood vessels at large. And we feel that it could also be applied to all these kind of indication. And that's how we have set up our upcoming research in the lab, including the 30,000, to test it in a corneal neovascularization models in mice. So the timeline, we have kind of in the next 6 months or so to do an ex vivo readout, basically looking at lesion regression, vessel regression in pterygia, but also do the mouse corneal neovascularization model. And building upon these, then we can establish the causation beyond the strong association and the mechanistic kind of knowledge that we have developed. And we feel the reason we built around the corneal neovascularization, there are corneal transplants failure have a huge economic risk, chemical burns, contact lens wear. Please don't sleep in your contacts. Please wear sunglasses. But so we feel there's an opportunity to expand beyond just pterygium. So thank you.
What technology did you use to identify your target? Were you using RNA-seq, proteomics? I must have missed that in the talk.
We use single nucleus RNA sequencing, so we try single cell RNA sequencing and single nucleus. And so both of these kind of modalities, we've used both of them on snap-frozen surgical specimens.
I noticed that this was a topical therapy. Would this be for healthcare professional application or patient application? And then what do you anticipate the dosing will be, and how often will it occur?
Sorry, I missed.
So would it be patient application or physician application, and then also how many doses will the patient require?
So I think options could be kind of quite open, depending on the mode of administration. It could be a topical drop. Obviously, for antibodies, there are also kind of dosing requirement. We can also easily inject subconjunctival injection, depot injection. So we envision different modes of injections or application, and it depends also whether we are using it to prevent recurrence or more for primary prevention, lesion regression. So it's a good question. I just don't. I'm not limiting kind of our options at this point.
And how many doses would you anticipate, or frequency of dosing?
So one of the things, if it is an antibody and it's applied, we have different kind of modalities. It'd be kind of up to 4 times a day, what is a lot of, let's say, ocular drops or eye drops. But there are also newer technologies because there's drop fatigue among patients. So the mode of administration is definitely something that we'll be considering.
So you're targeting the mediator. What's the phenotype of the knockout? Have you looked at that?
The phenotype of the knockout.
Yeah, you're targeting the mediator.
Yeah.
What's the impact?
Sure. Yeah. So I guess if we target the mediator, the downstream cell should kind of prevent this kind of ongoing self-reinforcing fibrovascular hemangiogenesis and angiogenesis that is characteristic of
in a normal eye. What's the function of the?
So in a normal eye, that specific mediator is very, very low. So it has also been so we are looking at kind of targeting the mediator and the downstream receptors. Maybe one would be sufficient, one not. There is an actual antibody developed against one of them in clinical practice and other indications, so that would provide a way to kind of repurpose it. Obviously, nothing in the eye, but it has good human safety profiles.
Great. That's all the time we have for questions.